A very good morning, dear students. I know you all are working very hard. I assure you, your hard work would pay. So keep on doing it. Today's lecture is about general embryology. I know it is a tough topic today, but what I need from you is concentration. Just concentrate, be attentive to today's lecture. It would be easy for you to understand. Okay, let's start. Today, what are the topics we are going to cover? Development of embryology. We are going to study the development of embryology. Not much, a superficial thing about germ cell and fertilization, formation of blastocyst and implantation, also the formation of germ layers. Then finally we are going to study about the differentiation, the formation of the Trilaminar germ layer, and how does the formation of the embryo takes place. So let's start. These are the certain names like Aristotle, Harvey, Malpighiac, Leeuwenhoek. They postulated certain concepts, and they have given us the information how the formation of a new individual takes place. Leeuwenhoek even described a micro-human in a sperm. Haeckel gave biogenetic law. Spearman gave the concept of experimental embryology. And who doesn't know about Dolly? Dolly, who created sensations all around the world, the sheep. It was a clone, see, in the University of Edinburgh, Scotland. Though died early as compared to its normal life expectancy due to lung diseases, severe lung diseases, and certain other diseases like rheumatoid arthritis. So this is about the development of embryology. Now the main thing which is required, you should know how to attempt in the examination. You can get a short note on fertilization. How to attempt? First of all, the definition. What is fertilization? Fertilization is a process in which a spermatozoon penetrates into the ovum to form a fertilized ovum. Yes, and this process of fertilization takes place in the ampullary region, that is the ampullary region of one of the part of the fallopian tube or the oviduct. You can see the sperms, they are deposited in vagina, and they pass through the uterine tube, and in the ampullary region sperm cell penetrates oocyte and results in fertilization. Now before fertilization, there are certain things you should know about the germ cells. Like there are two types of germ cells, spermatozoon and ovum. About the spermatozoon, it has to undergo certain physiological processes, that is capacitation. Now capacitation is the physiological changes in the spermatozoa to have the ability to penetrate and fertilize an egg. Okay, the sperm are enabled to bind to zona pellucida receptors. Also there is removal of glycoproteins, that is a decapacitation factor which cover the sperms. These glycoproteins have to be removed. Then what happens to ovum? Yes, there is formation of the secondary oocyte, and that is arrested at which phase? Metaphase, in the second meiotic division. This we already know. So if we just put together the steps of fertilization, there is sperm capacitation, sperm zona pellucida binding, acrosome reaction, penetration of the zona pellucida, sperm oocyte binding, there is egg activation and cortical reaction, and then there is the zona reaction. that is by the lysosomal enzymes of the oocyte. Okay, we can see the slide. This is an ovum, and see how many sperms are there, but only one sperm can fertilize the ovum. The sperm is traveling towards the ovum. Okay, this much we already know earlier. Regarding the oocyte, this formation of this secondary oocyte, and the second polar body is released and the nucleus remains haploid now. This is very important. Why it is important? Because haploid nucleus has to regain, the nucleus has to regain its diploid structure after fertilization. We don't want to increase the number, chromosome number, every time the fertilization takes place. Just to maintain the laws of inheritance and hereditary, the nucleus has to remain haploid. Okay. Now what next? What next? Let's see the slide first. How the contact of sperm and ovum takes place. Then is the penetration of the sperm into the ovum. Okay. Then you can see there is a formation of the pronucleus. Male and female pronucleus, they fuse. They have to fuse with each other. So there is fusion of pronuclei. Can you see the fusion? You can see the fusion of pronuclei. So the process of fertilization shows 4 main steps. The first is fusion of the membrane of sperm and ovum. Second is the nucleus of sperm penetrates into ovum. Then formation of the pronuclei. So there are the alterations of the ovum also side by side, and then there is the fusion of pronuclei. All these steps are within 24 hours of fertilization. You can see the fertilized egg also in the oviduct. It has to travel. Now what is the significance of fertilization? The significance of fertilization is there is the formation of a new life. New life. Then diploid number is regained. So inheritance as well as the aberrance, any abnormality, any deviation is also there. Yes, not the least one is the sex determination. Sex determination. So the fertilization, the formation of the zygote. The whole thing is that you can see from this slide, the secondary oocyte is released, the fertilization is taking place, then the formation of the zygote, then there are certain divisions, and finally there is implantation into the uterus. That is from ovulation to implantation. Now what next? How this, see, the things are not that easy. There is a formation of blastocyst. And what are the features? The first thing is the cleavage. There is cleavage, and then there is the formation of the blastocyst. The various steps are cleavage. Now cleavage is an early division of a fertilized egg. Then there is a formation of blastomeres. These are the daughter cells from the cleavage. There is a stage that is known as Morula. This Morula is about 16 to 32 cell stage structure. This reaches the uterus about 72 hours after fertilization, and it is still surrounded by zona pellucida. Then there is a formation of blastocyst. The name blastocyst arises from a Greek root word, means blast. I put forth shoots, and cyst means capsule. So the important thing is, the first thing is cleavage, And what is cleavage? It is a rapid mitotic. Please listen carefully. It is a rapid mitotic division of zygote to form a hollow spherical multicellular developmental structure, and that is known as blastula, and the stage is known as blastulation. This cleavage is a fractionating process. We can see the diagram. There is a division into 2, 4, 8, 16, 32, like this the division takes place, and this is the cleavage. Now this cleavage is since it involves the mitotic divisions, so the daughter cells, they are genetically the same. They are same or they are similar to the parent cell. And these divisions continue, these mitotic divisions continue until the average cell size, which is characteristic to parental organism, is achieved. And the daughter cells or the resultant cells are known as blastomeres. Now as the blastomeres, they do not move, so the size, volume and general shape of the blastula, it remains the same throughout the cleavage. Okay, the size, the volume, the shape, everything remains the same. And one important point over here is that oxygen consumption is very high during the process of cleavage. We can see the slide in which we can see the fertilized egg. Okay, then stage 1, that is there is the formation of the 2 cells. It is a 2-cell stage. You don't need to remember the R's. Then is the 4-cell stage. You can see there is the formation of the 4 cells. Okay, then is the 8-cell stage. So this is these are the phases of cleavage. You can see over here 12 to 16 stage, cell stage is reached over here, and that is known as the morula. Morula means mulberry type. So the up to at least 72 hours are required for the formation of the morula. This diagram is taken up from your books, in which we can see the various stages: the 2-cell stage, the 3-cell stage, the 4-cell stage. Then the early morula is formed. The trophoblast, the zona pellucida is still there. The zona pellucida is still there. Now the changes are that these cells, these blastomeres, these daughter cells, now they change. They form 2 layered structures, the outer layer and the inner layer. Outer layer is comparatively, the cells are comparatively small in size and they are known as micromeres, while the inner cells are large and they are known as macromeres. Zona pellucida still intact. Then what happens finally? This layer, that is the micromere layer, the outer cell layer, forms the trophoblast. The inner cell mass, that is of the macromeres, they are attached to one end and they are known as embryoblast. So trophoblast, embryoblast, and the cavity is known as the blastocoele, and this stage is known as the blastocyst. Is it okay with you? Is it fine? Is it clear? The main thing is there is differentiation of these cells into outer layer, which forms the trophoblast, inner layer that forms the embryoblast, and the cavity is blastocoele and the stage is blastocyst. This is known as blastocyst, from morula to formation of the blastocyst. So you can see the appearance, the inner structure. If you just, it is a ball-like structure. If you just cut it, it appears like this. Here is the inner cell mass attached to one area, one part. The same thing you can see over here, that trophoblast. Then you can also see the blastocyst cavity. Then you can see the inner cell mass. Same thing, the blastocyst cavity, you can see the inner cell mass and also the trophoblast. So this is a blastocyst, that is a hollow ball with inner cell mass. Yes, very good. Just study further now. The same diagram. Okay, inner cell mass, trophoblast and the blastocoele. Now what next? Now there is the formation of the embryonic disc. That is, this inner cell mass, it forms 2 layers of cells. and these layers of cells are separated by an extracellular basement membrane. This embryonic disc, which is formed by the inner cell mass. This inner cell mass now forms two layers. Please listen carefully. It forms two layers of cells. One is external and another is internal. The external layer is called as the epiblast. Internal layer is known as the hypoblast. Epiblast external, internal layer is hypoblast. And these two layers become distinct from day 8. Let's see now. This you are clear with the formation of the morula, that is solid ball of cells. Okay, at zygote undergoes cleavage. Then there is the formation of the trophoblast and the inner cell mass. This inner cell mass now certain changes. It forms bilayered structure, epiblast and hypoblast. Epiblast and hypoblast, and the two cavities, one on the superior aspect, another on the inferior aspect. This is known as the amniotic cavity. Below is known as the primary yolk sac. All these are by the formation of by the activity of the inner cell mass. Yes, good. Now the formation of germ layer. I'm just summarizing the same thing once again. It is the formation of the bilaminar germ disc, that is the inner cell mass, that is the two layers of cells in the disc, and there is the formation of the epiblast, which forms ectoderm, and these cells are columnar cells and they are adjacent to the trophoblast. And then there is the hypoblast, that is the endoderm. This consists of the cuboidal cells and they are adjacent to the blastocyst. Epiblast, hypoblast. Okay. See the bilaminar germ disc, epiblast and hypoblast. Epiblast consists of the columnar cells, hypoblast consists of the cuboidal cells. And you can see this is all by the, remember this inner cell mass? Inner cell mass. It forms the bilaminar, that is the germ disc, that is forms the epiblast and hypoblast. Columnar, cuboidal, epiblast, hypoblast. The cavity above, that is the epiblast, is amniotic cavity, and below the hypoblast is the primary yolk sac. We can see one more diagram in which we can see the epiblast, hypoblast, amniotic cavity and the primary yolk sac. This amniotic cavity is formed between the epiblast and the trophoblast. Above is the trophoblast, below is the epiblast. Trophoblast, epiblast is the amniotic cavity, and it is filled with the liquor amnii or amniotic fluid, and the roof is formed by the amniogenic cells. You can see these amniogenic cells, and floor by the epiblast. Roof is formed by the amniogenic cells and floor by the epiblast. While in the case of primary yolk sac, it lines the blastocyst cavity, and the membrane is known as the Husser's membrane. The membrane is known as the Husser's membrane. That is the amniotic cavity between the epiblast and trophoblast, and it is filled with this amniotic fluid, and its roof is formed by the amniogenic cells and the floor by epiblast. But for the primary yolk sac, it forms, there is a membrane, and the membrane is known as the, which lines the primary yolk sac, is known as the Husser's membrane. You can see this embryonic disc. Yes. What else you can see? The amnion. One more thing I can see over here, that is a connecting stalk. Connecting stalk is forming over here now. We'll study about this connecting stalk later. There are certain terms you have to understand. First, one is the extraembryonic mesoderm. Extraembryonic mesoderm, that is formed between the trophoblast and the endodermal cells. trophoblast and the endodermal cells, and this also separate the amniotic cavity from the trophoblast. Let's see the diagram, don't worry. There is a cavity which is formed, and that is known as extraembryonic cavity or chorionic cavity, or also known as coelom, and which this coelom, formation of this coelom results in the two layers. It divides the extraembryonic mesoderm into two layers, visceral as well as parietal. Visceral as well as parietal. Then there is the formation of secondary yolk sac and a body stalk. Secondary yolk sac and a body stalk. Let's see what we are talking about. So we talked about the extraembryonic mesoderm that is formed between the trophoblast and the flattened endodermal cells. Yes, it is forming over here, and they separate the amniotic cavity. This mesoderm is going to go further also. It separates the amniotic cavity from the trophoblast. This extraembryonic mesoderm is going to grow up over here, not shown here, but it is growing. It is in the stage of growth. Okay. Then what happens? There is the formation of the you can see extraembryonic coelom. Another slide we will see. This is a diagram taken up from books. We repeating the same thing once again. The embryonic disc, epiblast, hypoblast, amniotic cavity, primary yolk sac, and here is the formation of the extraembryonic mesoderm. Here is the trophoblast, and you can see this extraembryonic mesoderm is everywhere. This extraembryonic mesoderm separates the amniotic cavity from the trophoblast. Yes, this is a very simple diagram taken up from your books which you are studying for embryology. Then after the formation of this mesoderm, there is the formation of the extraembryonic coelom. This is the cavity. Cavities in the extraembryonic coelom is known as the chorionic cavity or the extraembryonic coelom itself. Now after the formation of this cavity, there are two layers. One is the parietal, another is the visceral. Parietal is also known as somatopleuric extraembryonic mesoderm or the chorionic plate, and visceral is also known as splanchnopluric extraembryonic mesoderm, which is outside the yolk sac. Let's see what do you mean by this. So we talked about the extraembryonic mesoderm. Then there is a formation of the extraembryonic coelom. Okay. Now this extraembryonic coelom, which is formed, it divides the extraembryonic mesoderm into two layers, a parietal layer and a visceral layer. Visceral layer is also known as splanchnopluric extraembryonic mesoderm, and parietal layer is also known as the splanchnopluric extraembryonic mesoderm, that is outside the yolk sac. Okay. Then also there is one more area in which this extraembryonic mesoderm is unsplit, and that is known as the connecting stalk. Chorion, amnion. Chorion is, please listen carefully, the parietal extraembryonic mesoderm. You know this parietal extraembryonic mesoderm plus trophoblast, that forms the chorion. Amnion is formed by the amniogenic cells, the parietal extraembryonic mesoderm and the connecting stalk. They together form the amnion. The secondary yolk sac is formed after the formation of the extraembryonic coelom, and it is smaller than the primary. Now your all queries will be clear if you see this diagram. Again taken up from your book. Very simplified diagram. We talked about parietal and visceral. Parietal means somatopleuric extraembryonic mesoderm, and visceral means splanchnopluric extraembryonic mesoderm. So here they are. You remember this amniotic cavity, you remember this yolk sac, you remember this disc, embryonic disc, the two layers, epiblast and hypoblast. Yes. Now after the formation of this coelom, The primary yolk sac becomes the secondary yolk sac. It is smaller in size also. Done. Then we talked about the connecting stalk. Here is the area of the connecting stalk where the coelom is not reaching. It is the unsplit part of the extraembryonic mesoderm. It is unsplit part. Then is the chorion. Chorion means the parietal or the somatopleuric extraembryonic mesoderm plus trophoblast. That forms the chorion. Amnion. Amnion is formed by these amniogenic cells. Amniogenic cells, the parietal, because it is not split, parietal extraembryonic mesoderm, and plus the, you can see the connecting stalk. All these three things together form the amnion. Okay, all the structures are clear to you? Easy to remember now? Now this we have studied about the formation of the coelom, the mesoderm. Now this disc's work is still incomplete. So we are going to study this embryonic disc once again. This much you know by now, that there are layer of upper cells that is towards the amniotic cavity, that is the epiblast, and that's the columnar. Then there is a layer of lower cells towards the yolk sac, and that is the hypoblast, and that is cubicle. Now this hypoblast, which has the cubical cells, some cells of this hypoblast becomes columnar, and they are known as procaudal cells, and together they form the procaudal plate. This procaudal plate formation determines the central axis of the embryo's head and tail end. The head and tail end should be known, so that is by the formation of the procaudal plate. But in one area, that is in the there is no infiltration of the mesoderm, and in this area the ectoderm and endoderm, they are in direct contact with each other, and that forms the buccopharyngeal membrane, that is the future mouth. See this diagram now. So what we can see in this diagram? The epiblast and the hypoblast, the columnar cells and the cubical cells. But certain cubical cells become columnar, and they form the procaudal plate. And this procaudal plate determines what? The head end and the head end of the embryo. So once head end is established, so the other end would be the tail end. Okay, that is the importance of the procaudal plate. So this is again a diagram from your book itself. Epiblast, hypoblast, and there is the formation of procaudal plate. Okay. Now the area where there is no infiltration of the mesoderm, where there is direct contact of the ectoderm and the endoderm, it constitutes a thin membrane because it has to break off for the formation of the future mouth. That is this membrane is known as the buccopharyngeal membrane or oropharyngeal membrane. And similarly, caudally you have the formation of the cloacal membrane. So buccopharyngeal membrane towards the head end and cloacal membrane towards the caudal end. There is no mesoderm. Then there is one more important term, that is known as the primitive streak. Now you can get a short note on primitive streak in your main exams. So please try to understand, and you should know what to write in this. It is a transient structure, and its formation approximately on day 15 of human development marks the start of gastrulation, and that is a process in which the inner cell mass is converted into the trilaminar embryonic disc. Trilaminar embryonic disc means there are 3 germ layers formation now. That is the formation of the ectoderm, formation of the mesoderm, and formation of the endoderm. See this? This is the inner cell mass. The embryonic disc is formed after the inner cell mass, yes, differentiates. Then this forms the 3 primitive streak, which give rise to 3 germ layers. And where is the procaudal plate? At the cranial end. And here is the primitive streak. Okay. Again, from your book, there is the appearance of a primitive streak. You can see in the axis. First of all, in the XY axis, you can see there is a formation of the procaudal plate and a primitive streak. This primitive streak and is formed this proliferation of the ectodermal cells. You can see the proliferation over here, and they have to move sideways. The primitive streak initially is round, then becomes linear, and there is a formation of cells. They proliferate along the sideways. They move along the sideways and they form the secondary mesoderm and the endoderm. You can see the slide. This is the primitive streak, and the cells proliferate, forming the mesoderm. You can see, and the mesoderm is of three types: paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. From the primitive streak. One more diagram. You can see the proliferation. The mesodermal cells are spreading sideways from the primitive streak. Here is the primitive streak. Now, the primitive streak is there, and the significance is that it determines the direction of the embryo and the formation of the notochord. Notochord we'll study later in the next class, but here the importance of primitive streak is that it determines the direction of embryo. Okay, so if we just summarize what we just studied, is that there is a formation of the bilaminar embryonic disc, which divides into epiblast and hypoblast, and epiblast forms ectoderm, primitive streak is there, and amniotic ectoderm. That is by the epiblast. E for epiblast, E for ectoderm. Then what about the hypoblast? Hypoblast, endoderm, visceral endoderm, hypoblast, visceral endoderm, and extraembryonic endoderm, which further this visceral endoderm forms the extraembryonic visceral endoderm. Hypoblast, endoderm, epiblast, ectoderm. You can see the formation of the endoderm. Epiblast, hypoblast, the primitive streak is there in between, and there is a formation of the endoderm. What about the formation of the mesoderm? Yes, this is the epiblast. Here is the hypoblast, and you can see the cells proliferating from the epiblast, and they forms the mesoderm. Epiblast to mesoderm. Clear? I hope. So this is the final result, that is the formation of the trilaminar germ disc. I hope the lecture is clear to you, and if you have any query, kindly let me know anytime. Thank you so much and have a nice day. Bye bye.